For the longest time, we've thought about age as just one number, the candles on your birthday cake. But what if that's an overly simple way of looking at things? Science is now showing us that aging isn't a single process. Instead, it seems to be many processes, all happening at different speeds throughout our bodies. We're essentially moving from a blurry black and white picture of aging to a high-definition, multi-system view. So this really gets to the core question that's changing how we understand getting older. And the answer, as we're now learning, is a pretty definitive no. Your heart, your brain, your kidneys, they all appear to follow their own distinct aging timelines. And that simple fact creates a major challenge for medicine. Because if we can't see these different clocks, how can we possibly predict a person's future health with any real accuracy?
So, to really appreciate how significant this new approach is, let's first take a quick look at how we've been measuring aging until now, and, more importantly, where those methods have fallen short. You see, we all really have two different ages. First, there's your chronological age, which is fixed. It's simply how many birthdays you've had. But then there's your biological age. Think of it like the mileage on a car. You could have two people who are both 50 years old, but one might have the internal mileage of a 40-year-old, while the other has the mileage of a 60-year-old. It's this biological age, the actual wear and tear on our body, that scientists are really trying to understand. And one of the most powerful tools we have for this are something called epigenetic clocks. A helpful way to think about this is to imagine your DNA as a huge instruction manual. As you get older, tiny chemical tags, or methylation patterns, get attached to different pages, kind of like little sticky notes. Now, these notes don't change the words in the manual, but they do change how the instructions get read. Well, epigenetic clocks are tools that can read the pattern of these sticky notes, which change so predictably over time that they can estimate your biological age from a single blood sample. Now, these clocks were a major step forward, but they have one pretty significant limitation. They give you a single, averaged-out number for your entire body. It's a bit like getting one overall grade for school without knowing your individual marks in math or history. It doesn't tell you where the aging is actually happening, whether your heart is aging faster than your liver, for instance. And that, it turns out, is a critical blind spot. And that brings us to this new framework called systems age. It was designed specifically to solve this problem, to finally let us see the individual aging clocks that are ticking away inside our different body systems. The researchers behind this work stated the problem quite plainly. They recognized that while the existing clocks were good at comparing one person to another, they were completely missing the crucial variations within a single person. So they created systems age to bring this internal aging landscape into focus. And what they accomplished is quite substantial. From just one blood sample, their method can generate a biological age score for 11 different systems in the body. We're talking about the heart, lungs, brain, the immune and inflammatory systems, right down to your metabolism. It's the most detailed picture of an individual's aging process that we've had to date. So you might be wondering, how exactly did they build this? Well, it was a complex, multi-step process that combined traditional medical data with some very powerful machine learning. Let's walk through a simplified version of how it works. First, they gathered standard clinical data, things like cholesterol levels for the heart system or grip strength for the musculoskeletal system, and sorted them into those 11 systems. Next, they used statistical models to find the core aging signals within that data. But here's the clever part. They then trained a machine learning model to find the DNA methylation patterns in blood that corresponded to those clinical signals. This created a DNA-based proxy for the health of each system. From there, they could build 11 separate scores and then combine those into one overall systems age. Of course, a new test is only as good as what it can tell us. So the most important question is, can these new specific scores predict specific diseases any better than the older single-number methods? Well, the findings were quite compelling. Let's take heart disease. The study found that their new heart system score was a stronger predictor of future coronary heart disease events than other available measures. This indicated that the test was indeed picking up on signals directly related to the health of that specific system. And this chart here really illustrates that point. It's comparing how strongly different aging clocks are associated with heart disease. The main thing to note is that a higher bar means a stronger prediction. And as you can see, the new heart score from systems age shows a stronger association than two of the leading previous clocks, suggesting it's a more accurate predictor for this particular condition. And this wasn't just true for the heart. This specificity was observed across the board. The brain score had the strongest link to cognitive function. The musculoskeletal score was the best predictor for physical issues like arthritis. And the blood score was the strongest predictor for future leukemia risk. In essence, each score acts like a specialized monitoring system for its own domain.
But beyond just predicting individual diseases, this research opens up the possibility of something even more profound: the ability to identify our own unique personal patterns of aging. The researchers are calling these aging subtypes.
Now, this is really the key concept. You can have two people who are the exact same age, and they might even have the same overall biological age score. But internally, they could be on completely different aging trajectories. One person's primary area of aging might be their metabolic system, putting them at risk for diabetes, while the other's is in their cardiovascular system, pointing toward future heart problems. This table from the study shows this perfectly. Take a look at these three individuals. On paper, their overall biological age is pretty similar, but when you look closer, you see individual A is aging fastest in their heart and lungs. For individual B, it's their metabolic and inflammation systems. And for individual C, the accelerated aging is concentrated in the brain and liver. Three similar people, but three very different aging profiles, which means three very different sets of future health risks. And it's these unique patterns that the researchers have formally termed aging subtypes. You can think of them as common blueprints for how different body systems can age, with each blueprint carrying its own distinct package of health risks. And this is where it becomes very practical for medicine. By identifying someone's aging subtype, you can begin to understand why they might be at risk for certain conditions. For example, the study found two different subtypes that were both at a high risk for heart disease. But for one group, the risk was clearly driven by their lungs and likely smoking, while for the other, it was driven by their metabolism and inflammation. Knowing that difference could fundamentally change how a doctor approaches prevention. So, how many of these aging blueprints did they find? In this initial study cohort, the researchers were able to identify 9 distinct aging subtypes. Each of these 9 groups shared a common pattern of aging across their body systems, and each pattern was linked to a different set of risks for future diseases. So let's just pull this all together. What systems age represents is a move away from a single blurry number for biological age and toward a much higher resolution map of our health. It provides a granular view across 11 different systems. It offers more specific disease risk prediction. And for the first time, it allows us to identify these personal aging subtypes. And perhaps the most compelling part is that all of this insight can come from a single blood draw, which could make it a very powerful tool for personalized medicine in the future. This line of research is pushing us toward a future where we might be able to know, with a high degree of precision, which parts of our own bodies are aging the fastest, which systems might be our weakest link. And that leaves us with a very personal question to consider. If that information were available to you, would you want to know?